to be with you tonight. I attended. I was telling Lois an event at the Atheist Society when we could meet all together in the same place, face to face. We seen that event was when Armin Navabi came, an Iranian dissident and atheist. Gave a really excellent presentation. So it's an honor for me to be able to join you and tell you a story that involves a discovery that I made in the Burgess Shale, a very famous fossil locality west of Calgary, and we're going to get into that. And so I can share my screen right now. And so let's see, let's go there. And there. And how does that look, Lois?
Well, we haven't seen it yet.
Oh, we haven't seen it yet. Okay, let's see. And let's go right there. Oh, it says screen sharing has failed to start, so I'm going to try again.
Okay.
Let's see. Okay, I'll go share. Sorry, guys. We tested this earlier and it was so good.
Yeah, it was fine.
Okay, let me go back and let's see here. Let's go right there. One participant can share at a time. So you're not sharing anymore, right?
That's right. So there shouldn't be.
Okay. Let's see. So you're not seeing my screen right now. Not quite sure how to fix that.
But try that now.
Okay. And I'll go right there.
There we go.
Yeah, okay, good. And I'll hit the play button here so we can get a large version. All right, I don't know what to call this talk. It's got a bit of a weird title, I realize, The Burgess Shale and the Three-Legged Table, because I want to tell you a little bit about this discovery in the Burgess Shale that hit the news, as Lois said, but also a little bit about my own sort of intellectual trajectory from, a pretty fundamentalist Christian when I first got to university, and now an atheist and a paleontologist. So this shot right here, taken by Miriam, my partner, when we were in Burgess Pass looking across to Mount Stephen, and we've got this radiant light coming through some clouds. In the field we call these God rays. And so we've got the God rays and the shoulder of Mount Stephen. And in fact, just in this little dark area right here is where I actually found these fossils that I'm going to talk about, some fossils that are new to the Burgess Shale. So let's see, here we go. All right, so just to get yourself oriented geographically, we're going to be talking about a couple, well, 3 mountains in particular. Now, for those of you that live in Alberta, at least, you will have driven by these mountains many times, because in fact, the #1 highway, if you see my cursor here, runs down here. And here's the little town of Field right here. And then you will have driven, if you've gone to, Vancouver or Golden or whatever, you will have driven right by Mount Stephen on your left and Mount Field on your right. And both these mountains host outcrops of rock strata known as the Burgess Shale. And I'll tell you about the significance of the Burgess Shale in a minute. And there's another mountain I'm going to show you a picture of along a ridge to the north of Mount Field, Wapta Mountain. All right, so when you are looking at Mount Stephen, you are looking at an enormous chunk of Cambrian time. So this is a time period going back more than 500 million years. I'll show you a time scale in a minute. And the whole mountain virtually is Cambrian rock. So right at the very bottom, now down here you can see that's the railway line. In fact, there would happen to be a train on it when this picture was taken. And there's the #1 highway right there. So when you're driving along here, you look up at your left, you are looking at an enormous chunk of Cambrian time represented by rock strata. I've circled that dark patch of rock right there because that is in fact where I found the fossils that we're going to be talking about. But that last picture may not have been that familiar to you unless you've been up in Burgess Pass. So if you ever stop in the town of Field and you look up to the big mountain towering above, that is Mount Stephen. And all of this is Cambrian rock. And this is where I found the fossils of interest, where the red circle is. All right, now looking at, to get ourselves oriented in terms of geologic time here, this is a very simplistic time scale here, but so we've got time on the right here and sort of a cartoon version of rocks and the kinds of fossils you might find in. So I put a green star here going way back to the Paleozoic era, and the Cambrian rocks we'll be looking at are about 505 million years old from the mid-Cambrian. So this is a really significant time in the geologic history of Earth, because it's a time known as the Cambrian explosion, when all the basic animal groups that we call phyla were first evolving and diversifying. So when we look at fossils in rocks worldwide, we find that they're in the same sequence. And so in fact, we can predict that sequence. People have studied, rock strata containing fossils, on every continent now, even Antarctica, and we find that the same sequence of fossils. For instance, these animals shown down here, the trilobites are always in rock strata below, for example, rock strata containing dinosaur fossils. And unless you have mountain building that are shoving layers up on top of one another, you will always find this same basic pattern worldwide. And so as a result, when you find fossils in rock strata, you can get a pretty good idea where you are in terms of the relative position in geologic time. Now you'll see over on this side, though, there's actually numbers. This came along later with the discovery of radioactivity and the realization that certain radioactive elements decay at a known rate. And so you can measure how much of the particular radioactive element is present in certain kinds of rocks, like volcanic ash, for example, and how much of the daughter product that the radioactive elements decay into. Uranium, for example, ultimately decays into lead. And so you measure the proportions of these, and so you can actually get a number of millions of years before present, and that's what these numbers are showing. Then the actual time for boundaries between these geologic periods. Now I know that from time to time we encounter individuals that look at this pile of rock strata and the fossils that are contained and go, well, these can be explained by a worldwide flood described in the book of Genesis, Noah's flood. And the notion being that why do we find things like horses and human remains and mammoths and so on at the top of the pile and not down here, is because as the waters are rising, these animals are going to go up to higher ground, and the smartest ones end up at the top, including humans. And so I don't run into this very often. Once in a while, students come by, typically from fundamentalist backgrounds. And so we have a chat. And I don't go into a lot of discussion in the class about this particular view of Earth history. I just make the students a bet. I've been teaching actually at Mount Royal University since 1998. And I make this bet saying to them, okay, well, rather than talking about dinosaurs and mammoths and humans running up hills as the flood waters are coming up, what about something like oysters? Oysters are pretty common. When you go to the seashore, especially along the west coast of Canada, there's oysters all over the place. They're around coastlines worldwide. So if oysters were present throughout the geologic, throughout the history of the world since the creation, surely we could find, for example, some oysters in Cambrian rocks. Now we've got this enormous pile of Cambrian rocks just west of us. And I make a bet with them, if you can find an oyster fossil in any of those Cambrian rocks, for example, on Mount Stephen or Mount Field, we'll see. Yeah, I will give you $1 million. And that's a very safe bet because you're not going to find any oysters there. Now, surely if oysters were around with trilobites and all the animals were created at the same time, surely we could find some oyster fossils in all those Cambrian rocks. And but we don't. Why? Because they hadn't evolved yet. They don't actually appear until the Triassic period. And so this sequence cannot be explained in terms of a worldwide flood. Sorry, go back there. Now, in terms of, I mentioned geography, but in fact, the geography was very different in the Cambrian. And so here's a view of the paleogeography of the world in the Cambrian, just a little bit after the Burgess Shale was deposited. I couldn't get one for the middle Cambrian, but this is a late Cambrian Paleogeographic map. Where's North America? Here. We call it Laurentia because Greenland is actually part of the continent and has been stuck with the rest of North America through pretty much all of geologic history. So this star shows where the Burgess Shale is, actually roughly where Calgary is as well. And of course, the continent, what is now the west side of the continent, was actually facing almost north. And certainly in the middle Cambrian, we were almost right on the equator. By the late Cambrian, we had drifted a little bit north. So in the Burgess Shale, then, we're looking at organisms that inhabited a tropical sea. Now, here's the classic place where the Burgess Shale was first discovered by a scientist known as Charles Walcott. This is, and you can book hikes here through Parks Canada or also through the Burgess Shale Geoscience Foundation. I guided that for them for about 10 years. And this is Wapta Mountain here. And so you can hike around, and there's a trail down here, and ultimately you hike backwards. And this is the Walcott Quarry. So this is a quarry that was started by Charles Walcott back in 1909. And more than 10,000 fossils were recovered through his quarrying and shipped to the Smithsonian Institution ultimately in the United States. And he wrote some classic papers describing the fossils. And the fossils are quite extraordinary, as I shall show you in terms of their preservation. The geology here is very interesting because you can see the rocks here are kind of gray. And then as you move to the left here, and I've made a yellow line where this contact is, they turn sort of orange. That's because we go from shales to a type of carbonate rock called dolomite. And this is an unusual relationship here because it's kind of a vertical relationship. And it's thought that this, in fact, represents a submarine cliff, the Burgess Shale piled up next to it, rather like stacking newspapers next to a wall. And so here's a little graphic showing you what the environment is thought to have been like at this time, with the Burgess Shale piling up next to a submarine escarpment made of this carbonate rock. And that is the man that discovered the Burgess Shale, Charles Walcott. So you can see he's got his, this is him in his quarry, and he's dressed for every occasion, even wearing a tie in the field, because I guess you never know who you're going to run into on the mountainside. And we know that the Burgess Shale is a big deal because it even appeared on a beer bottle label celebrating the 100 years since the discovery. A Big Rock Brewery put out Shale Ale with a picture, in fact, of Walcott and some of the animals of the Burgess Shale. So why is the Burgess Shale such a big deal? In fact, I doubt that you would find a geologist in the world that has not heard of the Burgess Shale. And so what makes it so important? Well, what's so great about it? Well, first of all, as I mentioned earlier, it's the great preservation of the fossils. So normally the fossil record is a record of teeth, bones, shells, and plant remains. But in the case of the Burgess Shale, we have soft anatomy preserved that is rarely encountered in fossils. And in fact, there are some animals that have no skeleton at all. This jellyfish-like creature known as a ctenophorine has no skeleton, and yet it's very beautifully preserved. Trilobites, for example, do have a hard skeleton, but they have rather lightly skeletonized legs that are almost never preserved in fossils, but preserved in extraordinary detail in the case of the Burgess Shale. So one of the great things about the Burgess Shale then is we can see aspects of the anatomy of 505 million year old animals normally not available to paleontologists. The second thing that's important about the Burgess Shale is its great age. There are some other fossil localities that preserve extraordinary details like that in the Burgess Shale. For example, in Princeton, BC, there are fossil lake deposits there that preserve insects, even the occasional bird, bat, fishes, even with some soft anatomy preserved. But probably you've never heard of the Princeton fossil beds, and most geologists haven't either. Why? Because they're only about 40 million years old, and a lot of the evolutionary action was over by then. But in the case of the Burgess Shale, we're going back to that interval known as the Cambrian explosion, when there's a lot of evolutionary action going on because the animals are undergoing their evolutionary radiation. And so this looks like a complicated chart here, but what we're looking at is this boundary between what's called the Neoproterozoic era and the Paleozoic era. And at that boundary, a lot of things happen. And what we see here is that, of course, the diversity of animals increases enormously. And the Burgess Shale then sits near the end of what we would call the Cambrian explosion, where we get this incredible increase in diversity. But you can also see this blue part of the chart here, where we have something called disparity. So that word's going to come up again a little later in the talk. And by disparity is something different than diversity. So maybe I can use an example like, imagine there's two furniture stores side by side. And you go in one furniture store and they've got 1,000 items on the display floor, but they're all bedroom suites. And so the diversity is quite high. We've got 1,000 items, but the disparity is not, because there's not a great range of forms. It's all bedroom suites. Then you go next door and it's a furniture store with 1,000 items on the display floor, but they've got bedroom suites, kitchen tables, couches, chairs, all of different kinds of furniture. So we would say that the disparity, the range of morphologic forms of furniture is much greater in that second furniture store. And so one of the things we see then in the Cambrian is an increase in the range of body plans in the animal kingdom. So we're going to come back to that concept in a little bit. Something else that we find is that we get an enormous increase in skeletonized organisms. Earlier on, back here, things like these strange organisms known as the Ediacara, they don't actually have mineralized skeletons. They were sort of soft-bodied organisms, but we start to see just the appearance of skeletons right at the very end of this interval of time, the end of what's known as the Precambrian, part of that being the Neoproterozoic eon. We start to see some skeletons appear, but these are very small organisms, only a few millimeters typically, and they have all kinds of weird shapes. So when we get into the lower Cambrian, we start to see an increase in the types of skeletons we see. And here's an example right here. We call these the small shelly fauna or small shelly fossils. And the animal I'm going to describe from the Burgess Shale would be, we would characterize as being part of the so-called small shelly fauna. So we'll get back to that in a minute. Now, I mentioned Walcott he made his discovery on Fossil Ridge. Now, I showed you a picture earlier of Wapta Mountain. and we have Fossil Ridge, and here's that submarine cliff right here. So you got to imagine that would be like a wall coming out of the screen towards you. The arrow shows where Walcott's Quarry is. And I got to just move this little window here. The discovery that I made was at the other end. And so had Walcott walked all the way to the end, maybe he did, I don't know, but he did not find these organisms that I'll be talking about, which actually occur right here, way at the other end. That's about a kilometer and a half. And so zeroing in then on the end of Fossil Ridge, where we come up to Mount Field, then we see dipping rock strata, and just in where this limestone is exposed in these trees here, I decided to take a look at the limestones, because the Burgess Shale, although the name connotates like the whole thing must be shale, in fact, it's not all shale. A lot of it is shale, but there are some limestone layers as well. And people tend not to look at the limestone layers because they're busy getting those beautiful fossils that I showed you out of the shales. And so I thought, no, people haven't really looked at the limestone. I'm going to take a look and see what's there. So I looked here at the L1 locality, and then across the Kicking Horse Valley, here's Mount Stephen, and there's that same limestone coming out on Mount Stephen. And so I went there, and I'll show you what I found. Oh, here's another view from Mount Stephen, looking back at Mount Field to show you where this sits in relationship to Wapta Mountain and Mount Field. And so this is me on that limestone outcrop. And looking very closely, I could see, well, in fact, there are some very tiny fossils. There's a penny for scale. And I realized that no one had actually described anything like this from the Burgess Shale, because I think that, they just hadn't really looked at the limestones, which surprised me a little bit because, the Burgess Shale has been studied for over a century. So I thought, this could be interesting. Now, the problem is, these tiny fossils, only millimeters across, how do we actually get them out of the rock to study them? You can see they're standing up in relief here because I realized, when I kind of got down there with my hand lens and looked very closely, the skeletal remains have been replaced by silica during their long entombment of, 500 million years, that very slowly carbonate ions are taken out of the shells and silica ions are deposited in their place. And as a result, I can take rocks like this and put them in acid, and the fossils are resistant to the acid, but the limestone around them will dissolve in acid. So I did that, and I got some very nice results. All sorts of interesting shells like those shown here. Now, some of these things I recognized, but there's one that I didn't, and that was that one right there. I thought, I didn't know what that was, a kind of an asymmetric shell. It's almost symmetrical, but not quite. So I didn't know what that was. And when I started looking at the literature, I realized it was an odd group of animals that have puzzled paleontologists for well over 100 years, known as Stenothecoids. And so what was great about the fossils that I found, I realized I had probably the best preserved ones known from the fossil record, because I was able to extract them using this acid technique. And so I was able to see internal surfaces and external surfaces. And so I thought, wow, okay, I've got some a lot of additional information here that was not available to paleontologists before me. So I thought, maybe I can help figure out, what kind of animals are these Stenothecoids, because paleontologists, as I will show you, have argued, where do these things fit in the tree of life? What even basic group we call phyla, where should they be classified? Now, I ended up naming, actually, this turned out to be a new species. I ended up naming it Stenothecoides rossettii, after Franco Rossetti. I want to talk a little bit about him, kind of a hero of mine. paleontological hero. I realized when I started looking at the literature that these Stenothecoids had been found on a number of continents. Here's, in fact, Franco Rossetti actually found some in British Columbia in Yoho National Park, even on Mount Stephen. So I thought, wow, he was working very close to where I found these Stenothecoids, but he was in rock layers well below the Burgess Shale. And in fact, in rock layers that probably would have been a couple million years older than where I found them. But the preservation available to him was not as good as the fossils that I found. So he wasn't able to make a definitive statement where they belonged in terms of classification. So we have some fossils from British Columbia of Stenothecoids, from Greenland, quite a few from Siberia, Mongolia, and China. And you can see they come in a number of different shapes. So what sorts of animals are these? All right, so now this is a rather complicated looking chart here, but let me just explain it. What I'm showing here is the history of interpretation of Stenothecoids, beginning with their initial discovery by none other than Charles Walcott, the discoverer of the Burgess Shale, but he didn't find Stenothecoids in the Burgess Shale. He found them in Nevada. And so in 1884, he described these shells that he found in Cambrian rocks in Nevada, kind of innocuous looking shells. And he decided that these were a type of snail. So what I've shown on the chart here is that I've got phyla, the basic animal groups listed across the top. So including the mollusks, which today include things like clams, snails, squid, and so on. And then I've got one here that you won't recognize, a Stenothecata, because as we shall see, one of the Russians decided that the Stenothecoids must be in their own phylum, completely separate from any other animal groups. The Brachiopoda, I'll show you what a brachiopod looks like. And even one scientist suggested that the Stenothecoids might actually belong in the Arthropoda, which includes things like trilobites, spiders, scorpions, and insects. So Walcott, he thought this was some kind of an early snail and suggested that, in fact, it was like a limpet. Now, you've all probably seen limpets if you walked around tide pools on the coast of British Columbia, these kind of cap-shaped shells, and the animal then is underneath, having the shell on its back, and this is a type of snail. So he suggested that Stenothecoids then just had this elongated cap-like shell on their backs and would have had a lifestyle rather like a limpet. So you can see, looking at this chart, that the general consensus was that Stenothecoids were some kind of mollusk. So everything that's blue, purple, and kind of greenish, these are all examples of scientists that interpreted Stenothecoids as belonging to somewhere in the phylum Mollusca. And of course, there's a lot of variation in terms of interpretation. We have one Russian scientist, Rozov, who decided in 1984 that he gave up trying to fit Stenothecoids in mollusks or anywhere else and said they must be in their own phylum. There were a few Russians, though, that decided that maybe the Stenothecoids belong in this phylum, the Brachiopoda, although they didn't really have a lot of evidence for that. And then this one scientist, Ressers, thought these were some kind of crustaceans. Okay, now a major advance came along with Franco Rossetti. Now, Franco Rossetti, a very interesting character. I'm going to tell you a little bit about him in the next slide. But he found in Yoho National Park, near where I was working on Mount Stephen, but in rock layers below the Burgess Shale, he found some Stenothecoids and published this in 1954 and 57. And here are some of the images. Now, he didn't have silicified fossils, so he wasn't able to dissolve up the rock to actually get these out as three-dimensional shells. He would just split with a hammer and then look at what was exposed. But he's the first one to actually show some internal features of Stenothecoids. So this is a view of the internal surface of the shell, showing these odd ridges around the outside, and then in the middle, chevron-shaped grooves. And so he thought that these must be some kind of muscle scars, and that this was essentially a segmented animal. So here's Rossetti here. Where did he place them? He thought, well, either in a group called the Monoplacophora, very primitive mollusks with a cap-shaped shell, again, on their backs, or the gastropods, which are a related group. But he wasn't really sure which class to put them in, but he certainly thought that they were in the phylum Mollusca. But a little bit about his life, because I did end up naming the species after him. Franco Rossetti, he lived to be 100 years old, born in 1901 and passed away in 2001. He was a physicist, and he was part of the Enrico Fermi's research group at the University of Rome. Here's Enrico Fermi on the right and Franco Rossetti next to him. And they worked on the concept of fission of radioactive elements, which ultimately, of course, became very important in developing the atomic weapons. So Franco Rossetti ended up fleeing the rising fascism in Italy in 1939, and he came to Canada, and he took a position at the Laval University, taught there for a number of years before being offered an opportunity to be a head of a physics department at Johns Hopkins University. Now they wanted Franco to work on the Manhattan Project to develop an atomic weapon in the United States, ultimately used against the Japanese. But he refused to do so on ethical grounds. And he became a little bit sort of disillusioned with physics just because he saw government intrusion and so on. And he became more and more interested in paleontology. And so he would take opportunities to come to Western Canada, to Yoho National Park, collecting trilobites. He wrote a number of excellent papers on the trilobite fossils and using trilobites to figure out where you are in the Cambrian time period. And so he also ended up writing a fairly definitive work on alpine flowers. So he was a real Renaissance type person. Okay, a big breakthrough came through with a Russian paleontologist, Setchev, in 1960, in terms of understanding what stenothecoids are. And he showed that no, these are not animals that have a kind of a cap-shaped shell on their back, but this was a bivalved animal that opens and closes its shell rather like a clam. And here's his images of some stenothecoids from Siberia, showing that there were two valves that fit together like this. And so he concluded, well, then these are bivalved mollusks, which of course bivalves include things like clams, oysters, and scallops. Then along came Robison, American paleontologist, and he found, he did find some silicified fossils in Nevada. And, and he showed some internal features here, and he said, look, there's a tooth at the top. And then he had some shells that seemed to lack this. So he thought, well, this is a tooth that fits into a socket, just like clams. And so he naturally concluded that the stenothecoids belong in the class Bivalvia in the phylum Mollusca. Now that happens to be a group that I study. I've spent quite a bit of my career on the evolution of the bivalves. And when I remember when I first read that, I was not happy with that explanation, because there's a lot of features of things like clams, oysters, and scallops that we do not see in the case of stenothecoids. Then we have Ellis Yolkelson in 1968, 1969. And he said, and he agreed that the bivalves were not the place for stenothecoids. And instead, he suggested that, that they belong in their own class. And he named that class the Stenothecoida. At the same year, another Russian named the group Probivalvia, but the name Stenothecoida has stuck. So here are some pictures of what he described. He only had a single specimen from Alaska, but he showed some good photographs of it. there's an end view of it right here. And he said, oh, look at this end here, it's broken off. He interpreted this as damage. This becomes significant a little bit later, as I'll show you. And so he actually suggested a reconstruction of the soft part anatomy as much as he could deduce. Okay, and finally, several times, beginning in 1937, some Russian paleontologists suggested a completely different phylum, the Brachiopoda. But as I said, when I looked at those papers, they really didn't have a lot of evidence, but they realized there were problems trying to jam the Stenothecoids in the phylum Mollusca. And finally, one Russian Rozov said, no, well, I'm just going to put it in a new phylum because it's just not fitting anywhere. Now, what's the problem with figuring out where Stenothecoids are in terms of, are they mollusks? Are they related to brachiopods? And we have a problem of symmetry. The brachiopods was very common in the fossil record. In fact, if you hike around Banff or Canmore up in the high country, looking through those limestones, you've almost certainly seen fossil brachiopods. They're very easy to recognize because the shells are absolutely symmetrical. They're bivalved, so they open and close like a jewelry box, but you can split each of the valves right down the middle and get two equal halves. And so we say that they are equilateral, or they have this bilateral symmetry that runs perpendicular to the junction between the two valves. Unlike the bivalve mollusks, which are a separate phylum, that evolved a shell that opens and closes like a jewelry box independently of the brachiopods, but their shells are symmetrical, at least early in their history. The two valves have equal convexity. They are unequally convex in the case of brachiopods. But if you look at an individual valve, you cannot divide them into two equal halves. So they are asymmetric. We say inequilateral. So they're equivalved, but inequilateral. The brachiopods are inequivalved, but equilateral. What about the Stenothecoids? How do they compare? Well, they don't fit either pattern because individual valves are asymmetrical like clams, but the shell is inequivalved. The two valves are of unequal convexity like the brachiopods. And so we got this problem of an animal that does not fit into recognized groups. So needless to say, that this created what I call phylogenetic havoc in the phylum Mollusca, because as I said, most paleontologists thought that the Stenothecoids belong somewhere in the phylum Mollusca. And so here's three different interpretations. We've got, let's see, some arrows showing you where these different interpretations place the Stenothecoids. So these are like what we call phylogenetic trees. And so Yolkeson thought he couldn't really say that they're more closely related to any particular group of mollusks, but that they were mollusks. And whereas Runagarum and Pagetta, they thought that the Stenothecoids were part of this group, the Triblidiids, which are have a cap-shaped shell, and they just said, well, the Stenothecoids independently evolved a bivalve shell, but actually belong within this group. Quite a different interpretation, certainly from Yolkeson. These guys, by the way, were my thesis examiners when I was a graduate student. And then finally, John Peel in 1991 said, no, you're going to put the Stenothecoids, he suspected some kind of relationship with the bivalves, which of course include things like clams, oysters, and such within the mollusks. So that's the consensus, that they're somewhere within the mollusca, but nobody knew where. Okay, so this is what I saw when I started looking at these closely. So I'm not going to go into too much detail here. It's a bit technical, but again, we have these asymmetric shells, and then this peculiar structure at the top, a kind of triangular shaped boss. The shell is inequivalved, one valve being, looking at it from the side, being a little flatter than the other. this structure, which was thought to be a tooth that fit into a socket on the other valve. Remember, Robinson interpreted this to be a tooth and then inserted into a socket on the opposing valve, rather like a clam. Turns out not to work. Why? Because it occurs in both valves. So this valve fitted against that valve, and those two triangular shaped structures abut one another like this. So it's not a tooth and socket type of arrangement. So what was Robinson looking at in terms of a socket? He was looking at specimens in which the triangular shaped structure that I call the apical boss had broken off and left a space. But I was able to show that, in fact, there's remnants of this structure and that this is an artifact of damage. So I found these fossils initially in 2001. I realized that's almost 20, well, that's more than 20 years ago. And to tell you the truth, I didn't know what to do with them when I first found them, even though I realized that they were very well preserved, better than what had previously been described. But I couldn't really add any new interpretation. That changed a couple of years ago, when I went looking for a project for some students, and I took out those trays with the Stenothecoids and looking at them again, and I tilted to a specimen, and I realized that there's an opening at the narrow end of the shell. And so here's one of the valves right here, and you can see, what's this thing? There's an opening. And then this is a very interesting specimen here. Looking at that narrow end, there's a big opening right there. And that's very nicely shown on this specimen. And then I realized I had one of those, that's interesting moments, that this is not a feature of mollusks. There are no mollusks that show an opening like that at the narrow end of the shell. But there is another group that does. In fact, now we go back to the Brachiopoda, because they do show an opening at the what I call the apical end of the shell for the extrusion of a structure that attaches them to the sea floor. So I thought, wow, I can show that Stenothecoids had an opening for this structure that's unique to the phylum Brachiopoda. And so I thought, I can now make a pretty strong case that, in fact, the consensus that these were mollusks was wrong. And in fact, those three Russians, going mostly by intuition, are actually correct in their interpretation that these are most closely related to brachiopods. And then looking on the internal surface, those chevron-shaped grooves that Rossetti had noted, and these narrow structures along the edges resemble structures that we see in undoubted brachiopods. So this is a drawing of canals on the inside of the shell of a living brachiopod. And so I was able to make some comparisons here and interpret these structures in terms of canals that we see in brachiopods. Somewhat different, no doubt, in the Stenothecoids, but at least now we had some good evidence for comparing Stenothecoids, not with mollusks, but with brachiopods. And then, to my surprise, when I started looking in real detail at the shells of the Stenothecoids, I realized that the silica had replaced the microstructural details of the shell, and it turns out to be quite fibrous, like brachiopods, not like mollusks. So here's a Stenothecoid shell under the scanning electron microscope. I was able to show a fibrous structure matching, here's a Cambrian brachiopod from the same rocks as the Stenothecoids, and you can see the similarity in that fibrous microstructure. And I was able to propose, once I realized that these were relatives of brachiopods, I contacted a colleague in Sweden, Michael String, who's a brachiopod expert, and he helped me, and we wrote a paper together that provided a new interpretation of Stenothecoids, and we were able to reconstruct what the feeding system was like and some of the soft part anatomy. And then an important job that paleontologists do is reconstructing the tree of life. And so I'm, of course, here looking at just a tiny little bit of the tree of life and trying to figure out, where do Stenothecoids fit? And so what we do is we construct a tree like this, showing major features that characterize branches. And so I ended up putting the Stenothecoids right here. These are a number of different fossil groups, and proposing how Stenothecoids might have evolved actually from a multi-plated cylindrical organism that reduced the number of plates to just 2 major ones. This is known from Morocco, and finally ending up with Stenothecoids. So in terms of, where this fits on the tree of life, I just showed you a tiny little segment of the tree of life, but here's what the tree of life looks like to paleontologists and biologists, based mostly actually on DNA evidence, actually comparing the genetic code of various organisms. And it's now recognized that there are 3 major what we call domains: the bacteria, archaea, and eukarya. Now you notice on this tree that some of the branches look very long, some of them quite short. That is an indication of the degree of genetic separation. And so, of course, in the case of the Stenothecoids, we're not looking at bacteria or these other single-celled organisms, the archaea. We're way over here in a tiny little branch within the Animalia. And of course, that's where we belong as well. And so, these individual branches here, of course, include thousands of species. In the case of animals, probably several million. And interestingly, this is a bit of an aside, but genetic evidence indicates we're more closely related to fungi like mushrooms than plants. So, you're more closely related to a mushroom than you are to an oak tree. And so we just want to zero in on where do the Stenothecoids fit in the broader branch of the animal kingdom. And so here is what that looks like. Now, of course, again, recall that, each one of these branches here actually includes, hundreds, if not thousands of species. And so we're interested right over here. Now, here's the classic interpretation of Stenothecoids as being part of the mollusks. And so what we argued was that that is incorrect. And so we ended up taking that branch and grafting it over here with the brachiopods that are part of a group called the Lophophorates. So that was our contribution. It might look like somewhat trivial, but that is a major job of paleontologists and some zoologists and biologists is trying to actually work out all the details of the tree of life. The amazing thing about evolutionary theory is that it shows that we're all related, whether it's sponges, jellyfish, hummingbirds, Stenothecoids, or whatever. We ultimately all share a common ancestor. The similarities are not owing to a common design, but owing to common descent. And so we're kind of a big family, but it's not necessarily a happy family because, of course, we do eat one another. Okay, now going back to the Burgess Shale, this concept of disparity in the Burgess Shale, and that is the range of body forms that we see. And so here's a great painting by Gurche, one of the artists at the Smithsonian Institution, showing a few of the Burgess Shale animals at the foot of that submarine cliff. A weird one here called Opabinia has 5 eyes on top of its head and kind of a vacuum cleaner-like claw coming out the front. And these animals here, Hallucigenia, actually shown upside down. It was not realized that, in fact, these stick-like structures are not legs, but in fact, spines that were on the back. This is not the head. The narrow end is the head. And with careful study, it was realized that these tentacle-like structures on the back here are actually paired structures and, in fact, are legs. So one of the things that we find out about the Burgess Shale is that there are a number of organisms that don't fit into established groups known as phyla, and phyla are divided into classes. And I showed you in the case of the Stenothecoids that they did not fit well either. So I've placed them, as a relatives of the Brachiopoda, but putting the Stenothecoids with the brachiopods, it expands what we call the disparity of that group, because as I showed you, and students aren't going to like this, because we learn as students in paleontology that brachiopods are symmetrical. And now we have a group that's asymmetrical. So we're expanding the range of body forms that we associate with the Brachiopoda. And there are many animals like this in the Burgess Shale that have features that just don't fit well into any of the phyla that certainly in modern oceans. Look at some of these weird things here, like Herpetogaster, that has a kind of a branching like structure here, a thick body, and then a little stem that attaches to the sea floor. This thing called Haploprentis, I don't know where you would put that. And kind of a fish-like creature here with a big opening at one end, and so on. So there's quite a few organisms like this in the Burgess Shale. Here's Anomalocaris, a very famous one that had two enormous claws at the front of the face. Now, this was noted by a famous evolutionary biologist, the late Stephen Jay Gould, who has written some incredible books, including this one called Wonderful Life, the Burgess Shale and the Nature of History, which I can recommend for some fine reading. And one of the extraordinary statements that he made in that book was, he says, regarding the Burgess Shale and this notion of disparity, the Burgess Shale includes a range of disparity in anatomical designs never again equaled and not matched today in all the creatures in all the world's oceans. That is a surprising statement because it's totally counterintuitive. So Gould is saying that in the Cambrian, you had a greater range of fundamental body architectures than you do even in modern oceans. Now that concept has been argued somewhat because people have said, well, what Stephen Jay Gould regarded as phyla, because he was willing to accept that this animal, for instance, Anomalocaris, was probably belonged in its own phylum. Now they're saying it's something that paleontologists call a stem group of the segmented animals, the arthropoda, that includes things like spiders and scorpions and so on. But that's somewhat of a just a nomenclatorial argument. But the fact of the matter is that Anomalocaris is really quite different than any subsequent arthropods. It doesn't even have legs on the underside. So this is the kind of image that I associate with my undergraduate training back in the early 70s. What does the tree of life look like in terms of its shape? And so, I learned that the tree of life had this sort of shape, of course, coming from a common ancestor and then gradually increasing in diversity and disparity through time. And we get greater and greater diversity going up to our modern times. And Gould said, no, that's not how it looks. It actually looks like this, a bush at the bottom, yes, having a common ancestor, but a whole bunch of branches early on showing what we call that range of body forms or disparity early in the evolutionary history of animals. And then we had quite a what we call a decimation at the end of the Middle Cambrian, where a number of these sort of basic body architectures went extinct. And then the few surviving architectures, we have about 30 phyla of animals still around now, more in the Cambrian, according to Gould. And then we have just really tweaking of these surviving body architectures through the rest of geologic history following the Cambrian. And a good analogy, I think, is something suggested, I think it was by a guy named Kaufman, who wrote a really good book I can recommend called At Home in the Universe. And he suggested that the Burgess Shale is telling us something about how evolution works. And that we can think about the evolution of bicycles as a kind of a metaphor. If you think about when bicycles first appear, there's actually a greater variety, a greater disparity, a range of anatomical designs of bicycles early in their history than there is now. Look at these things. You know, we've got some that had a little front wheel and a big back wheel, the penny farthing with a great big front wheel and a little back wheel. We've got, is this a bicycle? It's certainly not like the bicycle we think of now. And we have this, that's recognizable certainly as what we would call a bicycle. So, and then we have even these weird things called mono wheels, where you actually could ride on the inside of a big wheel. So what survived out of all these lineages of bicycles? There was decimation. A lot of these architectures just ended up to not be terribly functional, and these designs persisted. So a whole bunch of lineages, call them classes or phyla of bicycles, went extinct, and we've got these two remaining forms. All right, so now that was just a little bit about the Burgess Shale and kind of where that Stenothecoid story fits in and so on. And now I'm just changing gears completely. And I'm going to talk a little bit about my own sort of trajectory intellectually, beginning when I first started university as an undergraduate. So I got converted. I don't come from a religious family, but I got converted in about 1971 or so. And it happened when I went to, where did I go? I was at some park, and there was a big group there. They're called the Jesus People Army from, I think they came out of Seattle, led by a woman named Linda Meisner, and she had a buckskin jacket on, and you know, they all looked like hippies. So that's kind of cool. But they were, you know, really nice people. And you know, they talked about their conversion and how does it change their life and so on. So anyway, I ended up getting converted, and of course, very early on, I was taught, not having really had, I don't think, very good training in school in terms of understanding Earth history, how evolution worked, what the evidence for evolution actually was. So I became a pretty hardcore creationist. And I ended up going to university with that mindset. So the metaphor I use for kind of where I was intellectually at this time is like standing on a three-legged table. And so what supported my kind of worldview, a sort of God-centered creationist view of the world, was like a three-legged table, and the three legs holding up or propping up that view included the Word of God, like the Bible, and then what I call the handiwork of God, as the Bible describes it, you know, the creation, Earth and life and the universe. And then something else, the experience of God. I'm going to talk a little bit about that. You know, what is that? And so here's me standing on, you know, the Christianity table with these three legs propping me up. And when I look at sort of where I was intellectually, of course, we would use, you know, terminology like, oh, I'm free in Jesus. But in actual fact, in retrospect, my world was very tiny, circumscribed, just went to the edge of that table. And what's out here? The world, as we referred to it, you know, secularism. You know, some people say, well, this is the work of the devil out here. And you got to restrict your activities and intellectual pursuits to what we call Christian. So you might ask, well, you know, why did you even get into that? So I think that what attracts people to all sorts of different causes and so on, first of all, there's purpose. So what's the message? God has a purpose for you. And, God has a purpose for me. And you know, you're part of this community, and you're working for God, and it's kind of this purpose idea is very important. And what's that guy's name? Franco, the survivor of one of the death camps. he wrote a book, and he said the main thing that makes you happy is purpose. The other thing is community, that you're with a group of people that, you share values and, again, looking after one another. And that's a very strong pull as well. And then there's another component here that I call secret knowledge, kind of being in the in-group, that, you have knowledge that you actually know what's going on. And the world out there, they're not really in tune with it, but, you're in tune with it. I mean, Jesus himself even kind of used that sort of idea, telling his disciples that I talk to them out there in parables, but to you, I've given the keys of the kingdom, that secret knowledge. So, you actually know what's going on here. And so that's very attractive. I see this secret knowledge thing appearing in all sorts of groups and enterprises. I remember I was very poor as a graduate student. And so one of my friends was selling vitamins. And so he said, I should sell these vitamins, you can be in my kind of pyramid thing, make some money. And these vitamins are really good, And so he took me along to a kind of a meeting of the vitamin people, and people were standing up giving testimonials, how this had changed their life. And I thought, wow, this sounds like some of the stuff I heard at church, people standing up, giving testimonies about how Jesus has changed their life. Well, this was vitamins. And again, it's that secret knowledge that we know what the secret is to good health. It's these vitamins. And so I started seeing parallels with completely unrelated endeavors and ways of thought. All right, so then what happened to the legs of my table? They got kicked out one by one. The first thing that happened was problems with the Word of God. So I ended up, after high school, didn't really want to know what I do. So I went to a theological college and I started studying. I studied Greek because I thought, I need to be able to read some of these early manuscripts and so on. And then one of the things that really shook me up was that I found out there was all kinds of variants in these ancient manuscripts. And I thought, well, how could we have variants in the Word of God? Because I imagined, I think, naively that, God is dictating to the New Testament writers and the Old Testament writers, and they're writing down as they're hearing this. Maybe a rather naive view of it, but that's how I saw it anyway. And these variants aren't just spelling mistakes or slight changes. Some of them are pretty major. I remember one that actually did shake me up when I was at that college, was the story of Jesus and the woman taken in adultery, as in John chapter 7. That there's a consensus that was the earliest manuscripts of John don't show that story, and it's generally agreed that that's an interpolation that was added in the 4th century. I thought, the Word of God, somebody's sticking something else in there, and I thought, is God really a good communicator here? So that rattled me. And the next thing that happened was I went to university. I started off in pre-med, but I took a physical anthropology course. And I remember these skulls coming out and, in our lab being set in front of me, Homo erectus, Neanderthals, Australopithecus, and these things. And I looked at that and I thought, wow, how do I interpret this? What am I looking at here? These are human-like, but they're not really like modern humans. And so I realized, especially taking other courses in paleontology and geology, that the evidence for evolution was way stronger than I imagined. Because as a creationist, I did stuff like, I even ran the slide projector for one of the guys from the Creation Research Institute in California, a guy named Dwayne Gish. It came up to Edmonton to give a talk, and I ended up running the slide projector for him. And then when I actually took some courses and saw and started to understand the evidence itself, boom, there went my second leg of my table. And the handiwork, I realized, did not require a supernatural explanation. Now, but here's the big one, and this is a tough one, I think. Experience. Because I had an experience, the so-called baptism of the Holy Spirit, as it was called, which, included some weird things in retrospect, like speaking in tongues, and this experience, I still remember it. It was in this prayer meeting, and these prayer meetings were loud, and everybody's praying, and telling me just open up and just let the Holy Spirit in, and I'm praying away. And then, yes, I had an experience. It felt a bit like somebody kind of pouring warm water over my head. I went right through. A very kind of a peaceful feeling, I gotta say. And, it's gonna sound a bit weird, but I kind of saw an image of a set of eyes looking at me. And I can still visualize that now, even after all those years. So the question is, and you cannot tell somebody they did not have an experience, because you can't get inside their head. But what you can question is the source. And so for years, even after my 2 legs had been kicked out from my Christianity table, I still had that, what exactly did I experience there? And then I read an article in Equinox magazine. And there's the cover of it down here. Is it all in your head? So this was a story written by a journalist that went to see a researcher at Laurentian University by the name of Michael Persinger. And he was very interested in the neurological aspects of religious experience and some other things like alien abductions and that sort of thing. What is actually going on in the brain in these situations? And so I read that article, and the writer described, going to Michael Persinger, and I think the Michael Persinger, I think he had people in a deprivation chamber, maybe, which is nothing new, where you reduce all sensation, except that the difference was he put a helmet, a modified skidoo helmet on people's heads with a bunch of wires on it, because he could generate low levels magnetic fields around the temporal lobes. And so he did that, and then he would record, with a camera, people sitting with the God helmet on, as it's been called. And he noticed that people would tend to look over their shoulder during their time in the deprivation chamber, or sometimes he just had them in a chair. They would look over his shoulder. So later on, he would ask them, so, what about that? And people would say, yeah, I sensed a kind of presence with me. It was a nice warm feeling, that presence. Now, when the reporter in the Equinox magazine article went, as I recall, I've been looking for that article. I was able to find a picture of the cover. I haven't been able to get that article back. But as I recall, the reporter said that Persinger cranked up the magnetic field quite high, and this person had quite an intense experience, including visualizing a face. And so the explanation is that, as I understand it, and people have argued, about the validity of Persinger's experiments, because some research groups were not able to duplicate it, but others did. My understanding is that, the word that he uses is something called interhemispheric intrusions. So apparently, the seat of your consciousness is split somewhat between the 2 halves of your brain, and it's mostly in the left half, I call the big me. But there's part of you in the other half right here. And then the communication between these 2 sides is regulated by this bridge of brain tissue known as the corpus callosum. And as I understand it, the magnetic field generated by the God helmet interferes with the corpus callosum and the regulation of information going back and forth. So what happens is the little me says hi to the big me. And so when I read that, I thought, that makes sense in terms of what I experienced, that with the all of the environment of, where I was in this fundamentalist group, and then prayer and just let go and just let Jesus in, let the Holy Spirit come in and all that. And then there's music and there's clapping and even there's dancing and all sorts of things going on. It was pretty intense. That really this simulates so much stimuli that it ends up messing with your corpus callosum, and the little me comes over and says hi. That's how I interpret it. And so, and who knows you better than yourself? So it's part of you coming over, kind of giving your big me a hug. And of course, it feels like a warm presence. And in the case of the reporter for Equinox, the journalist, and he even saw kind of a face, and I had that. And then I started reading things like voodoo, for example. What goes on in voodoo ceremonies? And I saw there's a tremendous amount of similarity between various voodoo rituals and what I saw in the charismatic community. Sort of, ecstasy and people falling down and so on. And you could say, oh, that's all fake, but it's not fake. No, they're actually experiencing something there, I think. But of course, the explanation when I was in the Christian world was, well, that, voodoo, well, that's of the devil, right? But you know what? That didn't ring true. These people, this is part of their culture, and that didn't sound right to me. So anyway, that's how I interpret my experience side, that it was fundamentally related to stimuli, messing with my corpus callosum, and my little me, basically doing an interhemispheric intrusion on my big me. But it was not a bad experience by any stretch, but I don't think it was supernatural. What else have I got? Okay, so anyway, that was the end of my table. But you know what? I found out that the secular world is actually okay. And in fact, it was very freeing, because I realized the world was so much bigger than when I was just living on that circumscribed tabletop. So these things that happen, especially, when the handiwork table leg was kicked out by my experience as an undergraduate, ultimately in paleontology and geology. When I look back, my professors were enormously influential on me. And I had some, wonderful professors that were extremely patient. My very first paper that I wrote was on the evolution of chordates. And I talked about all these gaps on the fossil record that, can't be explained, that we have to have a supernatural agent to. So I can imagine when they were reading this, wow, this guy, we got a long ways to go here with this guy. But they were tremendously patient with me. They didn't try to change my mind all in one shot. They were patient. We had numerous conversations. They did not indulge in mocking. I don't think that's a good way to go. I think that would have alienated me. And all they did was, over time, they got me to question my assumptions. And they were just dropping in seeds of reason. And eventually that just sprouted. And so I ended up, abandoning completely my Christian roots, and I pursued a career in paleontology. I ended up getting a job. I studied in Australia for 4 years, doing my PhD, and came back to Canada when they were building the Tyrrell Museum. And so I was lucky to get a job there as a researcher in invertebrate paleontology. I was there for 20 years before taking a position at Mount Royal University. So, in terms of the God question, it's not really a question for me. I figure if there is a God that and I turn out to be completely wrong in how I look at things now, I think she will appreciate some intellectual honesty. That's all. Thank you.
Oh, thank you so much, Paul. That was fantastic. And I've got to say, I can identify so closely with your deconversion process. Oh, very much. Okay, thank you. So would you like to stop sharing the screen so we can all see
Yes.
each other? And
Sure.
I've got questions, but I think I will ask anybody that has questions to use the raised hand, and we can pick Paul's brain for a little while. Oh, wow. Okay. Brent.
I'm not sure which line to ask first when you get around to me.
Oh, well, I can see an order on the screen here.
Yeah.
Go ahead, Brent.
Curious, Paul, have you ever met a person named Stephen Godfrey?
I have. I believe he's at the Calvert Museum down in the.
Yeah, and he wrote a book called Paradigms on Pilgrimage. Stephen Godfrey is one of the co-authors. You might be interested in it because he has almost exact same experience you did. He came to university with a creationist view, but when he went to grad school at McGill into paleontology, he realized that his interpretation didn't stand on its own feet.
Name Godfrey.
Stephen Godfrey, yes.
Yeah, that's interesting because he was at the Tyrrell Museum for a little while. I think on contract back in the early days when it was first being built, and so he and I had a couple of very interesting conversations, and I know that he was already thinking about a book, but he was still working out a lot of his ideas and trying to figure out, his worldview was being adjusted right then and there. And obviously it sort of came to fruition with this book. So what's the copyright date on that book?
1995, I think.
95, yeah. So I would have known him in about 10 years before that.
Apologies, 2005.
2005, yeah, 20 years before.
So it might be, if you can find this book, you'd be interested.
I'm going to write that down. Paradigms on Pilgrimage. Interesting.
While you're writing that down, just an interesting experience, because I went to school with him at Bishop's, a small university in Leamington, Quebec, in a biology program, and he was a creationist at the time. So he did a paper on the Archaeopteryx, and at the end of the presentation, the professor said, "Oh, where do you fit this animal into the evolutionary tree?" And Stephen said, "Well, actually, I don't believe in evolution." Bang, the lights went out.
Yeah.
It was one of those kind of things where if you were superstitious, you'd think, "Oh, wow, that was weird."
Oh, I see what you mean. You weren't talking metaphorically there.
Well, yeah, so it was sort of, and we were in a room that didn't have any external windows, right? So right after he says, "Well, I don't believe in evolution," the lights go out, it gets dark. And so it was sort of like a lot of people knew he was a creationist and saying, "Oh, did God just intervene here?"
Yeah. So what year would that have been?
Well, that would have been 1981.
Okay. Yeah.
And then he describes, well, he went to McGill for his master's, and then he went for a year out at Tyrrell, field research. Yeah. So, I thought I'd just throw that out just for your reference.
I got to contact him. That's really great to hear. I've lost touch with him completely. I just knew he was, I think, at the Calvert Museum down in, was that North or South Carolina, is it?
Yeah. Well, actually, to clarify, he still has a sort of a quasi-religious belief, but he accepts evolution and the historical world being 4.5 billion years or whatever it is.
Right.
The other half of the book is a religious guy trying to reinterpret how he understands it in a philosophical level, the existence of God sort of thing. But the first part of the book you can relate to, I think, when Stephen talks about his let's call it his evolution of belief over. He was raised a young Earth creationist in an evangelical family, but then as reason took over, he moved away from that.
Yeah.
Anyways, that was my 2 bits.
Thanks, appreciate that.
Well, thanks, Brent. Okay, Jim, you're next. Unmute yourself, please.
Yes, there. Okay. Okay. I thought of a few things. Near the end, your talk about the God helmet stuff reminded me about a book that made a splash a few decades ago, The Origin of Consciousness and the Breakdown of the Bicameral Mind. Have you heard of that one?
Yeah, I think now I heard of it. I didn't read really anything about it, but I think from conversations and so on, was it the idea that your brain was more split early in sort of, going back to even classical times, and hence more prone to kind of superstitious or supernatural beliefs?
Yeah.
Is that the idea?
Yeah, the notion was that the communications from the other hemisphere were sort of interpreted as voices of gods.
Yeah.
Yeah. So I'm not sure how seriously to take it myself. But anyway, I was just reminded of it by your big me and little me stuff.
Yeah.
Yeah.
Yeah. I'll have to take a look at that. Yeah, the bicameral mind.
Yeah.
Hypothesis.
Yeah. But yeah. And then one thing about the Burgess Shale particularly. Now you looked at the end of the Wapta Ridge.
Yeah.
And then you did a little bit on the south of the Kicking Horse Valley.
That's right.
And then somehow I got the impression that more recently people went quite a bit farther south and found some more.
See, the Burgess Shale is not a locality. It is a rock stratum that goes through a number of different mountains.
Yeah.
And these soft-bodied fossils are turning up at a number of localities. So it's quite a big discovery the Royal Ontario Museum made south in Kootenay National Park, an area known as Marble Canyon near Stanley Glacier.
Right.
Yeah, you're right about that.
Yeah. And let's see, I have been to the Walcott Quarry, and, and I've hiked around Emerald Lake on the trail that goes maybe 100 meters below the Walcott Quarry several times. So that that view looking towards a big waterfall at the north end is fantastic.
Yes, you're looking over at Mount Carnarvon.
Yeah.
And that waterfall coming down that ultimately feeds Emerald Lake.
Right.
Yeah. So did you actually get into the quarry yourself?
Yeah, so I did that once. That was when I'd recently moved to Calgary in the 90s. I went there.
Yeah, because I guided for those guys for about 10 years.
So
beginning, though not until I think about 1999.
Okay.
98, 99.
Yeah. Yeah. So the time I did that, this was something with the U of C, and they got one of the park rangers to take us up from near the Tactical Falls. Yeah. So start from there because that's sort of the least elevation gain route in. Yeah. So is that the way you usually took people in?
Yeah, we call it Whiskey Jack.
Yeah,
go up to Yoho Lake and around the mountain. When we did a lot of our field work, I actually had some research grants, and we had to use helicopters because I had a field crew, and I had to haul up a bear fence, and we're camping for weeks at a time. Yeah. But lately, I've just been able to walk to these localities.
Yeah. Yeah.
Okay, well, let's keep going here. I think it's Gavin's turn.
Yes. Where is it?
Thank you. Paul, I've really enjoyed your presentation there, but there was close to the end where you said something, and I wanted to write this down because I thought you said it quite eloquently. It was something about the commonality of the species alive today is better explained by common ancestor rather than common creator.
Yeah, so that, of course, when similarities between organisms were noted going all the way back to Lamarck's time in the 1700s, it was thought that the similarities reflected common design.
Mhm.
And but as we came to understand evolution, we realized that the similarities are owing to common descent.
Yeah, common descent. Okay,
Yeah.
so it's better explained by common descent rather than common design, is what you
That's
said.
right. Yeah.
Just wanted to get that quote
Okay.
right. Thank you.
Okay, thanks, Gavin. And Joel?
First of all, I'd just like to thank you for the presentation. The one thing that I found really interesting was your analogy for why there was more diversity at the beginning. You used the bicycle analogy, which I thought was really good. That was one thing that in the past I've wondered about, and that explained it very well. I've just got a side question. If I go hiking around around field and in the area, if I find fossils, can I keep them, or is that restricted? What is there any restriction on that?
Yeah, there is. So the way it works in a national park, you're not allowed to remove any natural object without a permit. So you would have to apply if you want to collect fossils, you probably wouldn't get it unless you're affiliated with some kind of research group and that the specimens are going to end up in a natural history collection somewhere. They don't do it because you get a pretty hefty fine. Now, if you're outside the parks in Alberta, I'm not sure what the law is in BC, you can pick up any fossils that are on the surface and take them home and have a collection, but you can't sell them and you can't export them out of the province without a permit. And you can't excavate. So if let's say you found a dinosaur skeleton, you can't bring in equipment, pickaxes and shovels or front-end loaders or something without a permit. You can pick stuff up off the surface that's already kind of weathered out of rocks.
So, okay, yeah, thanks.
Yeah.
Okay, and I think Andy has a question. Is that right, Andy?
Yes, thank you. And I'm new to the group, but a wonderful talk. Thank you very much, sir. I was interested in your explanation at the end of the sort of rapture that Christians and other people talk about. I read a book and that it's little me talking to big me. There's a book, and I think I read it about 10 or 15 years ago, called Why God Won't Go Away. And my understanding of that was that there is a part of our brain that is sort of, when it's stimulated, we will receive sort of transcendental thoughts, pleasurable transcendental thought. Is that linked? Is those sort of ideas, the idea that you proposed and that idea that I understood from why God won't go away, they're similar sort of explanations.
Yeah. Now, I haven't looked at the Persinger stuff for quite a few years, but as I recall, he was also looking at trying to identify what he regarded as a religious center of the brain that with certain kinds of stimulation, like you might find in a Pentecostal ceremony or maybe a voodoo ceremony, that you actually get a micro seizure, like a micro epileptic seizure in that part of your brain that induces a kind of ecstasy type of experience. I do recall reading something like that. So, but you know what? Yeah, that's getting out of my field. All I attempted to do was try to explain, kind of how I understand what I experienced, and also my intellectual, what I would regard as an intellectual transformation. Which, by the way, always gives me hope that, I know sometimes, you run into people and you go, man, where do I begin? And it's a waste of time even talking. Don't think that, because my professors could have done that with me. And I don't know where I would be right now, actually. But that patience and so on, was in retrospect is very inspiring. And so it gives me, a motivation and so on to, when I run into the people that maybe had views like I had and so on, they're not a lost cause by any stretch. But I think it takes, those professors, boy, they had skill. I don't think have you ever heard of something called street epistemology? Yeah, those guys, I think, were doing it and they didn't even know it.
Thank you.
Yeah.
Okay, thank you. And Jenna Lee.
Hi, hi, Paul. Thank you so much for that great talk. Sorry, we were a little late. And I just also wanted to give a shout out to Miriam. I know some of you were at our Skepticamp last year, and Miriam spoke at our Skepticamp. So nice to see you both. And I don't know if you spoke about this right at the beginning, because I was a little late, but we are actually planning a field trip this summer to Burgess Shale, and Gavin maybe is starting to think about how we organize that. But are you still leading? Are you still guiding?
No, I let my my permit expire. So what I'd be happy to go with you. We would book it probably, with the Burgess Shale Geoscience Foundation. It costs, I think, something like $70. Now, there's two places you can go. You can either go to Mount Stephen, the trilobite beds. I've done a lot of research there. In fact, I wrote a field guide for that deposit. And then also the Walcott Quarry. That's a lot longer day. That's about 11 kilometers in and 11 kilometers out. Whereas Mount Stephen, I think it's about 3.5, no, maybe 6 or 7 kilometers up and 6 or 7 kilometers back. It's a shorter day, but it's steeper. Yeah, that's definitely, that's a great idea.
Yeah, well, actually, I think when Brent and we went out to Tyrrell Museum last summer, and Brent suggested that we look at that for this summer. So we're going to organize it for July. Mm-hmm. So we'll let you know.
Okay, cool.
Okay, thank you. Jim, I see you have your hand up, but Stephen hasn't had a chance yet. So let's let Stephen ask his question first.
Okay. Well, thanks, Paul, for a very nice lecture. Learned a lot about shale and fossils. Towards the end, you talk about the three-legged stool. And one of my colleagues at Mount Royal, actually she teaches at the nursing school. And she ran into problems with students who remained on the three-legged stool, because that's the way the student was brought up. Especially one student, she mentioned that she came from a rural community and went to a very small school, very religious oriented. So all her life was brought up under the influence of creation. And her aspiration was to help people, so she wanted to be a nurse. And when she came to Mount Royal and attended the biology class, it was a total shock, because my colleague teaches evolution, which was totally contradicts to what she was brought up with, okay? And unfortunately, the student remained on the three-legged stool. And my colleague said, actually, she ran into trouble with students like that because they would argue in class about creation versus evolution. Now, Paul, in all your years of teaching, have you ever run into situations like this?
You know, surprisingly, not a lot, because I think some students come in probably with worldviews like this, but of course, in a geology class, as opposed to a biology class where they got to teach some physiology and they got to teach, circulation and nerves and all that stuff on top of, I suspect the evolutionary component is probably a rather small component of it. Whereas in a geology class, I have time to kind of set up, how rock strata form and, the evidence for a very ancient Earth. And I think that because I'm able to assemble this bit by bit, that the questions kind of percolate away. A lot of it's answered as we go on. Yeah, I've had the odd question, but I certainly haven't had any kind of, even in my 20 years there now, I've never really got in any kind of an argumentative situation, surprisingly. But I think it's just a function of the different structure of the class and the kind of time I have to build this hypothesis up for the students.
Yeah.
I'd be happy to talk to that student if they ever wanted to chat.
I'm sure she'll have a disagreement, because one of the things she thought was the Earth was 6,000 years old. Yeah. And you try to convince that with the Cambrian period and it's 4.5 billion years old. That's a big disagreement in those kind of situations.
I don't run into it a lot, surprisingly. Maybe a lot of the students, they're just being quiet and, kind of wrestling with these ideas, kind of within. Now, Miriam and I ran into a very strange worldview, at least strange from our vantage point. Just a couple of weeks ago, when we went to see a performance called Shen Yun.
Yep.
And we didn't realize that it actually is a kind of a performance promoting Falun Gong.
It is. They advertise themselves as China before communism.
Yeah.
But it is actually, I think the group was based in Taiwan. Falun Gong was very strong in Taiwan. They were promoting their religion as well.
Yeah.
Yeah.
But we went, and the dancing was absolutely fabulous, just fabulous. Let's turn on my light here. But then some strange things started happening. They had kind of a big animation behind the stage, and they showed these kind of, mystical people coming from space to Earth and so on, that humans are kind of some kind of galactic or godlike beings that came to Earth and have some kind of mission or purpose. And then at one point, there was a soloist and singing in Chinese. So I didn't know exactly what he was saying, but a giant banner appeared on the screen, that's like 20 meters across, and it said, atheism and evolution are deceitful. And Mary and I looked at each other like, wow, where's this coming from? So, and afterwards, when we went out to the lobby at, I guess it was an intermission, they had a camera for a TV station in New York. This dance troupe comes out of New York, apparently. And so they wanted to interview, what do you think of Falun Gong and so on? So I said, yeah, no, I'm happy to be interviewed. And I told them, I said, look, I teach evolution at a university, and I'm an atheist, and I don't think I'm deceitful. and I don't know why you felt like you had to push that message there. And then
they are actually banned in many places, actually. Because they are almost like a cult.
Yeah.
And I'd like to see this summer, actually at the Calgary Stampede Parade. Every year when we had the parade, there was actually a big, huge Falun Gong band there. They
Interesting.
all wear bright yellow clothing. And they do drum and gongs. And it was a very large, in fact, a large marching band. They are a very powerful group actually in the United States, and it's almost a cult. You got to watch out for them. I remember they have people standing in front of the Chinese consulate in Calgary, and there was like persistent demonstrations with banner, and people will kneel in front and have their hands like the Buddhists pray. So these people are very, very, very delicate, very extremely dedicated. They are kind of very out there. I would say kind of dangerous.
Yeah. Anyway, it was a shock for us because, and of course, it was coming from a completely different direction than fundamentalist Christianity. You know, but the parallels were interesting.
Yep.
Yeah.
Wow. Thank you.
Thank you.
Okay, Bonnie, would you like to ask your question?
Sure. Thank you, Paul, for that really interesting talk. The whole from the beginning to end. My question to you, as a professor, do you find and see that the belief in evolution is increasing overall, or is it going sideways, or is it decreasing?
I think that it's increasing overall. I think that I think that the high school education is deficient in actually explaining it, especially in the context of Earth history. There's no Earth science that's taught in the high school curriculum in Alberta. And so, you know, I see the students coming in, you know, they have no concept really of geologic timing, except what they picked up, because, you know, a lot of them, you know, at some stage are interested in dinosaurs and so on. Few of them been to the Tyrrell Museum, but overall, I think the training in Earth sciences is really poor. They get chemistry, biology, and physics, but Earth science integrates all those things to give us, you know, an understanding of the history of Earth and the history of life. But overall, yeah, I think it's probably increasing. I think that, you know, sort of fundamentalist religion, I suspect, I don't have any data on this, but just from my interaction with students, you know, I would have to say I'm kind of seeing it somewhat less over the last few years.
Okay, there's hope then.
Yeah, there's hope.
Thank you.
Jim, would you like to ask again?
Yeah, this one should be short. What's the latest in fossil record that these
Stenothecoids
show up?
Now, the ones that I found are among the youngest geologically. So they appear in the early Cambrian, and they went extinct in that late, at the end of the middle Cambrian, where you had that decimation of a lot of, you know, the fundamental body plans. We don't really know what happened there geologically, but yeah, so they had about a, I would say, a 25 million year history.
Okay, so we can't pick up any on the beach today, and
if you do, let me know. I want to see that.
and have a stenothecoid.
I'll give you $1 million. Yeah.
Okay, Stephen.
Oh, my hands up. Oh, just one very last one. Paul, actually, in your field of studies, all these layers of rock and things like that. I know I drive across the Rockies, and I saw these different layers, different colors. How can I tell? Actually, like how old they are? Is there any way, if I were to just park my car and look at Castle Mountain or look at the shale and say, this is so many billion years old? Is there a quick way?
Well, yes. If you get a geologic map, and then you find where you are on the geologic map, and you'll see that the geologic maps are very colorful. They look quite complicated, but if you can find where you are, let's say you're in something that's, you know, a patch that looks maybe blue, and then you go to the legend, and it'll tell you all the blue rocks are Devonian in age. And the Devonian, you know, is around, you know, 370 to about 400 million years old, rocks of the Devonian period. So that's one way to do it, is when you're traveling, actually have a geologic map with you. You can get a lot of it online. And there are books as well, quite nice geological highway guides that can help you to know what you're actually looking at. You know, when you look at Castle Mountain, by the way, that whole thing is all Cambrian as well, except the very bottom is you're going even deeper into time into what's called the Precambrian. And also when you're at Lake Louise as well, when you're looking at Lake Louise, all the cliffs around the whole lake are all lower Cambrian. So it's even older than the Burgess Shale, and it's mostly sand, not shale. So it's coarser material. And that actually represents the final kind of infilling of what was a rift that occurred when Antarctica and Australia split away from what is now the west side of North America. And as it's splitting away, of course, it leaves a great big hole, you know, like a rift valley that starts filling up with sediment. That's what you're looking at when you're at Lake Louise.
So like I would have some sort of a chart, like a laminated sheet of paper chart. I can just look at this and compare. Hey, I'm looking at this because I have no clue, and I always like to find out what it is.
I don't know if I could do it just with one chart. It probably require a few pages, but yeah, you know, I might be able to help you there, Stephen, to get some resources for when you're traveling through the Rockies.
That would be great.
Handbook of the Canadian Rockies.
Yeah, that's the one by Ben Gadd, eh?
Yeah.
Okay.
Yeah.
Handbook of the Canadian Rockies.
Handbook of the Canadian Rockies. Yeah, I think I have it on a shelf behind me here, actually.
We probably have it in the library. Okay, thanks.
And Dee Dee, what would you like to ask?
It's not really a question, it's more a comment, and it's kind of out of order, but it has to do with what Bonnie asked. Today, it's making shockwaves around the homeschool Christian community, particularly in the US. The homeschooling curriculum called ATI from the Bill Gothard Institute has just completely collapsed. So the student that Stephen was referring to in Alberta might even have been educated under this homeschooling curriculum that is, you know, Christian creationist propaganda, but it is completely, as of today, fallen, and they're not using it anymore. So that's good news and progress.
So when you say fallen, what do you mean? What do you mean fallen? You know, is the Bill Gothard Institute like no more? Is that what that means?
Well, he's under investigation, for instance, and there's a documentary being made about him right now, and the Duggar kids were all educated under this curriculum, and so there's been a few things happen. And when I say fallen, I don't really know what I mean, except for this is new information today. And watching homeschool TikTok, it's really like Christian kids who were educated under this curriculum are now just like they knew they had a bad education, but now Bill Gothard Institute, the IBLP, I think is the acronym, they're now saying, we're not promoting this anymore. We're not updating it. It's,
Wow. Yeah.
this is new information, so I'm not really sure of what you pay attention.
Yeah.
It's a pretty big deal.
Oh, thank you, Dee Dee. That's good information.
Hopeful.
Yes, very. Well, I'd like to throw in a couple of things here. Do you know who the most difficult people to deconvert are? The theistic evolutionists.
Oh, yeah, sure.
Like I was, because just put them together and you just keep doing mental gymnastics on everything, and
I tried that for a while too.
yeah,
You end up with almost a schizophrenic view of the world.
Tell me about it. Yeah, but I'd also like to tell you about one experience. Speaking of the experience situation, where you know that you've had some kind of experience. I was almost completely deconverted, but I was going to some group therapy, and one day all the emotions broke out, and the way things were happening, all of a sudden, to myself, I said, "This is an altar call." And all of the emotions that were going on in that group session that day were exactly like the emotions in an altar call.
Mm.
And it was just so fascinating to me. And I realized that human touch and belonging and being accepted are natural human needs, and that churches and altar calls usurped those needs and used them.
Yes, and that, like I said, you know, when I look back, so what was the draw for me? Well, the community side of it was a big thing, and that's why I was impressed by the your website. That I thought it sort of projects a notion of community and kind of looking after one another, and that's a really important element.
Thank you. It's what we are hoping for.
Yeah.
Yeah. Jenna Lee.
I just want to ask Paul if you think I'm going to be arrested, because then I don't even know, you won't be able to see these very well. So I have a whole like bowl of these. These came out of from my family farm in about 1911 when they dug the dugout.
Yeah.
But they're from Saskatchewan and now they're in Alberta. Am I in trouble?
No, I don't think so. I think the law is fairly similar in Saskatchewan. Those look like baculites to me.
Yeah, I think so.
Yeah. Which, by the way, in Indigenous cultures in southern Alberta, those are referred to. Yeah, they're quite iridescent. The beautiful mother of pearl. They're referred to as an iscum, and they end up in a lot of medicine bundles. Yeah. So baculites, for those of you that aren't familiar with them, are fairly common in parts of southern Alberta from shales from the Cretaceous period, when there was a seaway going through the core of North America about 75 million years ago, all the way from the Gulf of Mexico to the Arctic Ocean. And you had various animals, you know, living in this interior seaway, including baculites, which had an elongate cone-shaped shell and a squid-like animal protruding out of the wide end. And so their fossils are fairly common in parts of the Bearpaw Formation, which extends well into Saskatchewan.
I've got a bunch of crinoids from Nahahi Ridge. I don't remember how what the geologic age of that is offhand.
That would be Mississippian,
Okay.
and so that would be around let's see, 373. That'd be around 350 to 360 million years ago. From the Mississippian period. Yeah, there's a lot of these crinoid bits in these Mississippian rocks, especially in Kananaskis country.
I've got one more, well, sort of idea question. when you were going through the stages where you were talking about how you learn things, and you know that one part of the organism told you something anyway. The
Yeah,
idea is how research is done. And I have sort of, in my own mind, worked out an idea, and I want you to evaluate it, that if once you learn how research is done in any field, like my research was pretty much biochemistry, then at least you understand the concept and it helps you believe the science.
mhm.
Is there any way we could apply that idea to the current anti-science? You know, well, how you can't really know, but if you know how research is done, then you can accept it.
Yeah, I know what you mean about a little bit of an anti-science sentiment that seems to be evolving right now, coming from a couple of different directions. Not only, people have lost some trust in science, mostly around medical issues, because a lot of medical research has been shown to be, they haven't been able to replicate it. And that's because there's tremendous pressure on research groups that are being funded by drug companies and so on to, get the results that the drug company wants. There's a lot of money at stake. The other area is climate change research, where there's a tremendous amount of grant money out there, and I have colleagues that we think there's tremendous amount of hype around the whole climate change issue, and especially the way it's portrayed in the media. I don't want to get into that right now. That's almost the subject of another talk, but yeah. But the thing is about science is that unlike some other endeavors like politics and religion, evidence will ultimately win out. And it is, sometimes it takes a while, but it is ultimately self-correcting. Whereas, and you're not necessarily trying to hold up a view and resist all criticisms and so on. You're trying to defend this edifice, but instead it corrects itself over time. And a graduate student can overturn the biggest idea of the most well-established scientist somewhere if you actually have the evidence.
Yeah.
And so the appeal to authority doesn't ultimately work in science.
Right. Yeah, that's why I keep thinking if you need to understand how research is done. Yeah. And it helps you accept it better.
And even, my interpretation of those Stenothecoids, that could be, I might be incorrect in some aspects of that. I'd be happy to see how I am incorrect, quite frankly. So what I do is, like most people in science, including yourself, you look at all the data, the evidence you have, the observations, you try to propose a hypothesis that explains everything that you can see. And then somebody else might come along with new data that shows, now actually you need to alter that, that hypothesis, and that's just how it goes.
Oh yeah.
Yeah.
Well, that's the self-correcting part of it.
Yeah, and so I haven't proved that Stenothecoids are actually part of the the Brachiopod phylum versus the mollusk phylum, but I think I can say that we can have more certainty about my hypothesis than previous hypotheses. So science doesn't prove things. You only arrive at greater and lesser degrees of certainty. Yes. Math has proofs.
Gavin is grinning because he loves statistics, and he's going to be giving a webinar on thinking statistically.
Interesting.
Anyway, Andy, did you have another question?
Thank you. And I just wanted, just further to the question on acceptance of evolution, do you not think right now we're going through a dramatic lesson on evolution with the various strains of COVID? Is that not as traumatic a lesson as we could receive?
Yeah, I think though the problem when you bring that up, is that viruses are hard for most people to kind of relate to. You can't even see them unless you have a scanning electron microscope. And in fact, they're not actually living because they don't metabolize. They're in that nether world between living and non-living, which I talk about actually in some of my classes, because the notion that there's something intermediate between living and non-living is kind of a mind bender. And the student, we have great discussions about this because you can have a jar of virus and it doesn't consume any oxygen, doesn't do anything until it ends up inside of you, kind of like a monkey wrench being added to some complex machinery. But yeah, you're right. If you think about, the number of people that have got COVID around the planet, millions, how many virus particles were produced in each human? Millions among millions of, so you multiply millions times millions times millions, and the chances of you getting some mutations with all those replications and the little mistakes, and then you end up with a variant that has a different outer protein configuration, and now we have to, our immune systems have got to figure that out.
No, I agree. That's a good example.
Yeah, but evolution.
A quick example.
Yeah.
The viruses are the undead.
Yeah, I like that. I'm going to use that.
Does anybody else have any more questions? This has been fantastic. And okay. David.
Sure, Paul. That I really enjoyed that. You mentioned a book by someone Kaufman, and I didn't write it down. Could you?
Yeah, I forget his first name, but it's Kaufman, and it's At Home in the Universe.
At Home in the Universe. Okay, thank you.
Joel, can you mute yourself? Joel, thanks.
Okay, super. Thank you.
Okay. Well, Paul, I could sit and talk to you all night. I'm just so fascinated. This has been absolutely terrific. And I do want to know if you are aware of one particular program that we have in ASC, and that's our Life Without Religion peer support group.
Mm-hmm.
And again, community and mutual support is really important in people that are deconverting or have recently deconverted.
Yeah. I think that's a really important aspect of what you guys are doing. And while I'm actually, from looking at your website and so on, I've become more interested in your group. And I'm probably going to join, I think. Maybe I have already. I don't think so.
Absolutely.
But,
super.
Ooh, another convert. Oh my.
Convert.
You're like...
Yeah, I like the secret.
We like those converts. Yeah.
Yeah. So, do you have you said there's going to be a talk by was it Andy?
Gavin.
Statistics.
Yes.
Thinking statistically, yeah, that's cool. When's that coming up?
Let's see what. It'll be the last week in June. Have we come up with a date, Gavin? I think we have.
It's May 26th for that.
Oh, I'm sorry, May 26th. Super, thank you.
Statistics and decision-making under uncertainty. So kind of ties into what you're saying of science doesn't prove things definitively, but you start to get more and more certainty. So
yeah, that's right.
It ties into that.
Nice.
Super. Okay, well, again, we can't thank you enough, and we'd be thrilled, absolutely thrilled if you would join us, and because we want to create a soft place to land for people that don't have a place.
That's really well put.
And that's one of our big goals.
Not that we converts, no one.
Yes.
We're deconverts, aren't we?
We're deconverts. Yeah.
In one of the recent Skeptic's Guide to the Universe, they were talking about you have to give the person an alternate worldview rather than just debunking their current one.
Yeah.
Mm-hmm. Okay, well, thanks again, Paul, and this
All
has been
right.
terrific.
Thank you.
Thank you.
And hopefully we'll be seeing you again sometime.
Absolutely. Yeah, yeah, it was a
Yeah.
lot of fun. I appreciate it. Thanks so much.
Yeah. Paul, I
Okay.
may be seeing you at school.
Yeah, well, let's have a coffee here, okay?
Yeah.
Okay.
Oh, Stephen, are you the one who invited him? No.
Hey, Paul,
Okay.
Good.
Okay.
Paul, before you leave, I put in a link to that book that's now online from Stephen Godfrey.
Okay,
So
Okay.
I'm gonna just check the chat here. And Miriam noted that it's Stuart Kaufman, Stuart, at Home in the Universe. Okay, and the link to,
Thank you.
okay, I see it.
Yeah,
All right,
that's-
everybody,
Okay.
take care.
Okay. Take care.
Talk to you
Okay.
again.
Good night.
Good
Good
night.
night.
Good
Good
night.
night.
Bye.
Thanks again, Paul.